This is the beginning of the lecture information for Exam 4. This portion of the course follows very closely the textbook, the Campbell textbook. So I'm going to go a little faster, I think, in here because Chapter 23 and Chapter 24 follow very closely what it is I'm going to be talking about, and I'll be referencing an awful lot of figures taken directly from those chapters. Before we get going into Chapter 23, though, we really want to pause here at this portion of the course where we've finished taking a look at Darwin's major idea, along with, of course, Wallace, natural selection. We've learned and read and been tested about the implications, really, to his ideas. And so what we have to do, I think, at this point, before we go on, is really think about the significance of Charles Darwin and Charles Darwin's work. So what was it that we gained from this special insight and the application as well? So both Darwin and Wallace thought of the ideas, but Darwin was really the person that went forth and applied it and said, well, here's really what it would all mean. Okay. What Darwin was able to do was provide an explanation for biogeography. Why is it that organisms in certain parts of the world are more similar to each other than they are to organisms that live in similar environments far away? Why is it, as we looked at it, that organisms that live on islands are most like the nearest mainland? Why is it that ocean islands that are very, very far away, that is very far from a coastline, a mainland, maybe 1,000 miles, like Hawaii, why is it that they tend not to have natural populations of amphibians? These were just some of the biogeography that we looked at. And with Darwin's ideas, we were able to come up with some explanations. Darwin was able to help us understand the fossil record. So some of you on lecture exam 3, the essay question at the very end asked, what was it that's in the fossil record that's inconsistent with a 6,000-year-old world? And so one has to understand, well, okay, that's what the question was. Here was the creationist hypothesis that there was a worldwide flood and that simply what we find in the fossil record is jumbled together rocks and fossil evidence of organisms. But in fact, that's not what we find. Your possible answers for that essay question are almost endless. For one thing, if things settled out so that they were not evenly distributed in the fossil record, then you might expect things that are the heaviest would be at the bottom. So perhaps the bones of dinosaurs would be in the lowest levels of that rock. Well, we know that's not the case. Perhaps things that floated might be up near the top, whatever that would be, if it would be logs or wood. Is that what we see? No, it's not what we see at all. Instead, what we see are many of the points that we made about the fossil record. And so I'm not going to go through all those again, but it is that we find ancestors as we go deeper, and that we find really those common ancestral forms for major groups as we go deeper. So we see a loss of diversity as we go deeper, and we see a convergence as we go deeper of a few common ancestral forms. We also see that all of the rock layers do not contain all of the different types of organisms. We have very distinct places where trilobites ended. We have a very distinct place where non-bird dinosaurs ended. Remember that layer of iridium rock. Well, why would that be? Why would iridium settle out in just one layer? And when it settles out, you only have dinosaurs below. These would have been great things to bring up on the answer to that question. And these are the things that Darwin was able to explain with the fossil record. We understood, and this is a question that I had raised in zoology when we started thinking about this, we understood that life is clumpy, that life is not an even continuum of forms, but instead there are clear groups. There are things that are mushrooms. There are things that are like oak trees. There are things that are like butterflies and beetles and earthworms, and they're their own group. Remember that we don't have blurring between a dog and a tree or any other kinds of organisms in these groups. We don't have half dog, half mushroom, whatever that would look like. Okay? We have very distinct groups. And so the question was always raised, well, how do you explain these distinct groups? And one was a supernatural creator had an idea about a mushroom and made a lot of different types. And that's supernatural, and that's not testable. That's not predictable. It doesn't help us to really understand the world going forward. Instead, a scientific explanation is perhaps the members of a group are united by a common ancestry. Perhaps the members of a group are really a family in the broadest sense. That's what Darwin provided. We know that whole fields like comparative anatomy and embryology, some of you might be taking development this fall or in a year and a half taking comparative vertebrate anatomy with me. In both of these, we will look at those lessons, those evidence of evolution. Why is it that we have the 1, 2, and then lots of little bones and then rays of bones in the arm of a human, in the arm, the front leg of a horse, in the front flipper of a dolphin, why, or a turtle, or a salamander? Why do we have that pattern? That is both a pattern we see in the adult anatomy as well as in embryos. So we looked at lots of evidence in comparative anatomy and embryology that is best explained from an evolutionary perspective. And finally, why would it be then that members of groups, so Linnaeus's groups over here, when we are looking at Linnaeus's groups, why is it that then we find that not only do mushrooms look alike, mushrooms have much more similar molecular biology than they do with other kinds of groups, like beetles. Mushrooms are clearly their own group, separate from beetles, and mushrooms versus beetles have two different types of molecular biology. Well, these things are explained if you understand that they're family groups, that in the same way you came from your parents and grandparents, extending back, of course, literally thousands of generations, that these organisms have shared traits like a family. So the molecular biology of mushrooms is much more similar to other mushrooms than it is to beetles, and beetles in turn are more similar to each other. Not only did Darwin explain these things, he provided the mechanism by which life changes, and of course that is natural selection. And remember the old unity and diversity of life. The unity of life is the common ancestry, and the diversity of life is how natural selection can cause and lead to changes, how changes accumulate. Of course, those are adaptive changes, changes that arise and which convey advantages to the members of the surviving species. So, that's huge. What's on the screen right now is giant. It is as big as you will get in all of biology. Clearly, evolution, as provided by an awful lot of Darwinian mechanisms and insights, is the biggest idea by far in biology. Darwin's work was the synthesis. And thus this quote from Theodosius Dobzhansky, who was working about 100 years ago, helping to provide the synthesis of genetics and evolution when it was coming together in the neo-Darwinism, which is neo being new, which was a new perspective on Darwin's ideas by integrating genetics. That's the quote from Dobzhansky that we had on our syllabus. So when we think about famous scientists and their important contributions, it's not because they found something, they discovered a fossil, or they discovered a molecule. It's because they had mechanisms, they had a synthesis. Einstein was coming up with ideas that was a real synthesis, a real masterful, very, very encompassing idea. And Darwin is that to biology. And that's really important to understand. I think people still, and you can go, of course, to the internet and find casual or perhaps heated, somewhat of a debate, if you want. I don't think it's a debate, where people will disagree. They feel as if they can just toss to the side evolution, as if they can just accept or not accept evolution. And I wonder if those same people think that they can accept or not accept gravity or other characteristics that we know well in science, the characteristics of the world, of biology. Some of the people in our government were quite resistant to the threats posed by the COVID-19 disease. SARS-CoV-2 is actually the virus, as I understand it. And so some people were saying, no, it wasn't going to be a big deal, or maybe it was a conspiracy, that it was somehow a political kind of thing. And here's my point. No, these are science, facts of science. These are things that are just out there. And choosing to ignore something that is reality, well, that's not the world that we're trying to live in as biologists. We're trying to live in a natural world, not a make-believe world. And so this horrible virus is ravaging our country. The numbers are escalating. It is a catastrophe now, when you're reading this, close to the deaths of 9/11 every day. Today, it was announced that 2,000 American citizens died in the last day. And I understand that at 9/11, that less than 3,000 people died. And so we are approaching a 9/11 loss of life every day. So these are the real things. Life evolves, physics happens, people who seem that they can just dismiss these things are not helping. That's my point. And in fact, if we have people in government who don't understand evolution and who don't understand the physics that Einstein taught us, well, those people will place us at risk. And so that's why I get quite excited about people who deny science. It's a real concern. So we have to ask this question. What sort of world would we live in if we didn't understand Einstein's work? And what sort of world would we live in if we choose to ignore Darwin's work? I've been trying to follow the science closely. I'm not a microbiologist, but I certainly can read science articles and try to learn more about these things. And I understand that there were 2 recent studies done of the SARS-CoV-2 virus that causes COVID-19 disease, and that when they looked at that virus in New York, they found that it actually was most similar to the virus not from China, but instead most similar to the virus from Europe. And so in New York, stopping planes from coming from China might have been helpful for other areas of the country, but not for New York. Okay? These are the real life things about the science and what we're dealing with today. So you're majoring in science. It's an unbelievably important field, and in this national emergency, it is critically important. Those of my former students who have gone into the health professions make me very, very proud. Many of my recent students have gone into nursing. All of those students in medicine and all of those students in laboratories that are helping to contribute to addressing the ravages of this horrible pandemic, they know that science is going to be the way that they're going to move forward. Why? Because it says it right here. Science helps us understand our world. We can make predictions, and hopefully we can make this a better place for those of us that get through this. Right now, what we're wondering about is how that virus is going to change. So I mentioned to you that in New York City, the virus is actually traceable back to Europe. Well, probably how? How did they determine that? They determined it probably by mutations or changes to the virus. And that's right, the virus is evolving. And there's a concern that if you've been infected with the virus, that people might think that then you're going to be immune to it. But what if it's evolved? And what if next time around when you're exposed to this virus, that whatever immunity you built up from the last time that you were exposed is no longer effective? Yes. Now give some thought to that, because this is really important. People are making assumptions that after you've been through an exposure of the virus, or maybe even had the disease, that you somehow will be immune to it and can go back in the workplace. But is that true about the flu? Is it true that if you had the flu this year, that next year or 5 years from now, you won't get the flu again? Well, you know that that's not true, because we get flu shots every year. Why? Because the flu virus evolves. And now you're understanding why I'm spending so much time on this. An understanding of evolution, an understanding of the importance of science to make predictions about how this disease is going to evolve helps us to figure out how to work. Many of us are wondering, when will life return back to the world that we knew a year ago? And we understand that nobody really knows, because we don't know the traits of this virus. We don't know if having an exposure means you're resistant in the future. There's evidence that for those of you that experience very few or maybe no symptoms at all, that whatever immune reaction you had may not be sufficient to fight it on exposure in a month or in 6 months or a year. We don't know. We don't know if any of the medicines that we're trying right now are going to be effective. But darn it, we're going to use science to try to figure out if they are. We're not going to use hunches or feelings that were expressed by our president. What we're going to do is use science. And Dr. Anthony Fauci has been saying that all along. We're going to wait until we get the data, not hopeful thinking. It is important to be hopeful. But goodness, I hope that we rely on the science and the evidence about which drugs are effective at fighting this disease. And I am most hopeful that science will reveal some drugs that are effective at fighting the disease. I understand that drugs could be effective in 2 different ways. It might be something that you take to avoid being infected. So you might be able to be on some medicine if you're in a high risk area, like a hospital worker who's dealing with patients with COVID. And the second kind of drug category might instead be something that really helps you recover for those poor people who are maybe needing or on a respirator now. And there's lots of hope in that. But my hope hinges on using science to show us what are better approaches or strategies for this. And you need to know that this virus is evolving. And when you hear people make reference to the virus as being smart or clever, or the virus figured something out, the virus doesn't have a brain, and you'll remember that evolution is not deliberate. Instead, what's happening is there are situations that are favorable to the virus, and that's where we will have more virus. And there are situations, conditions, that aren't favorable for the virus, And we know that means we'll have less of that. If the virus varies and some variation conveys advantages for viral replication, then that's what we will see more of. It's not because the virus figured something out. It's because variation arises through random processes, and then if it conveys advantages, we expect that there will be more down the road in the days and months and years ahead. All right. Well, this big section of the lecture has much more meaningfulness for us today. So in a broader sense, beyond simply this disease, why do we need to understand evolution? So we just discussed fighting the flu or fighting SARS-CoV-2, the virus leading to COVID-19 disease. We have discussed before fighting antibiotic resistance. So we're dealing with a virus now, but antibiotic-resistant bacteria are another huge threat and an altogether different kind of organism to fight. We've talked before about dealing with pests that are hurting our crops that are helping to feed the world. We've looked at this before. You need to understand evolution so that you understand what organisms would be good model organisms for human medicine. I don't know if you've been following the news close enough that some of the hopeful drugs, the drugs that show promise against COVID-19, some of those drugs have been tested on organisms that seem to get COVID-19, or some kind of model system that helps us make predictions about what might be good in us. Understanding evolution helps us understand related species. So it might be that although a certain large group of cats living in a region are doing fine, one particular species is threatened, perhaps by a virus. Well, if we understand that the cats in that region are similar enough to each other that they might catch the virus. I've now been aware that there's concern that great apes, naturally living great apes, Africa, we have them in Asia, that those great apes could also get infected and have COVID-19 disease. Why? How? Well, because we're closely related. Evolution also puts us in perspective, and that's how we will end this class. So I'm beginning right here, the lectures on exam 4 information, and then we'll have a test over that. And then we'll have a rather short series of lectures that will really wrap things up, and that'll be for lecture exam 5, and that will be given during the final exam where we have a comprehensive final. So in the very, very last exam 5 information, I will be trying to put us into this evolutionary perspective. And for those that need a real practical kind of thing, yes, it's on the exam. Evolution is on the MCAT exam, and an understanding of evolution is expected for people who are going into medicine, at least into medical school or a DO school, podiatry school, where you have to take the MCAT. People entering medicine need to understand the theories, atomic theories in physics, and they need to understand evolutionary theory just as well. All right, so now we're ready to start moving forward. I had made some reference to neo-Darwinism, or what is sometimes called the modern synthesis. The modern synthesis is that. It's a synthesis in modern times, about 100 years ago, of genetics and Darwinian ideas. And I said Theodosius Dobzhansky, who gave us the quote that's on our syllabus. Theodosius Dobzhansky was one of the people that did that. So let's take a look at the genetics involved in evolution, or looking at genetics from an evolutionary perspective. I'll say off on the side that across the United States, many courses in evolution that are addressing evolution spend an awful lot of time on genetics. And in fact, there are places that just simply marry the two together. And so evolution and genetics is the course. Here, it's something I'll talk about here in chapter 23. I'm going to use this whole lecture to get through chapter 23, so it'll be a long time. And I've got a little bit of coffee here. I think you'd expect me to sip a little coffee as I talk about these big ideas. In any case, evolution and genetics is the subject of chapter 23. So we're going to go through a little bit of genetics as we move ahead. Well, one of the important things to think about is the genetics of what? So as we look at evolution, what is it that evolves? Do individuals evolve? No. And this was a difference that I had pointed out in zoology class, in which I distinguished between development and evolution. And in one version of the word development, yes, you can think about change and change over time. And that version of the use of development could be something of a synonym for evolution. But development really is the changes in an individual lifetime. An individual lifetime. This bird develops. But this bird is not the unit of evolution. Natural selection does not work on individuals because individuals don't vary. They're an individual. Individuals within a population vary. So no, individuals can't evolve. What we looked at with natural selection was variation within a population. Okay. So we'll hear some misunderstandings about individual evolution. We've talked about this before. The change in individual, we said, is called development. Okay. Or in response to environmental change. So an individual will develop and an individual will respond to its environment through acclimation. I don't know if you remember some of what we said before, but if you go and live at a high altitude for a few weeks, the density of red blood cells inside your blood will increase. That's acclimation. Okay? There are things that an individual will do in response to environmental circumstances. Individuals also develop. Natural selection's acting on these individuals, but it's populations that evolve. Okay. So, when we talk about genetics, we usually talk about population genetics. That's the kind of phrasing that we're going to be talking about. Variation, genetic variation within a population. Okay? That's what we're going to talk about moving forward. We've said before, if there's no variation, then how can there be selection for any one type? It would seem that if there's no variation, then the population as a whole will either be successful or not. And we talked about little variation in the case of asexual reproduction that we looked at in zoology. And if you can remember, it was towards the end of the class, where did we see asexual reproduction near the end of the class? In an organism that switches between asexual and sexual reproduction. What did we look at that switched between asexual and sexual reproduction? And maybe you remember that that was aphids. Well, when things are going well, and in fact this time of year right now, early spring, plants are coming up with new vegetation, and aphids are, it's just like opening a buffet for aphids. Okay? And so aphids, when the going is very, very good, like early spring and summer, then aphids tend to reproduce asexually. Things are going fine. all those clones, all those little identical babies are going to do just fine. But we said variation arises during times of stress, and variation is produced by sexual reproduction in aphids. So these variations provide alternate strategies or alternate combinations of traits, or maybe even new traits that allow the aphids to do something. So first, variety, inherited variety, randomly generated variety, and then selection. That variety is the variety within a population. Variety within a population. Okay, so before we go any further, we've got to remind ourselves about some basic terminology in genetics. I think it's very likely that you had this terminology in high school, but it doesn't hurt to have a little refresher before I start using terminology, and you might not be clear about what things mean. So what are genes? Well, genes, and the definition of genes, has changed over time. Genes are units of inheritance. That's pretty much how we define a gene. Genes usually influence a single trait, but we now know of exceptions. Lots of organisms are diploid. We're going to talk about some exceptions to that. An awful lot of the things, the fresh fruits and vegetables that you may have trouble getting right now because you might not be going to the grocery store as often as you used to. In any case, lots of fresh fruits and vegetables that you're eating have more than 2 of every type of chromosome. Okay, so 2 of every type of chromosome. That's what it means to be 2N, or diploid. Sex chromosomes are the one pair of chromosomes that are not necessarily identical. Now, of course, in women, usually you have 2 X's, and in human males, it's an X and a Y. Alleles, and this was, I don't know, in my education somehow, I didn't pay attention to this definition, so I was confused. I also, when I was growing up, didn't have good textbooks around me, and so I couldn't look these things up. Now you guys can just punch it into Google and get a definition if you go to a good, reliable source. So alleles are just variations of a gene, and you can have some fun with this. You can, instead of a gene, G-E-N-E, you can think of genes as in blue jeans, J-E-A-N, and you can think about going to a store and seeing if in the store they have different kinds of pants, different kinds of blue jeans. Maybe there's some that have more rips than others. Or imagine this, some blue jeans that don't have rips in them. Those are the kind that your parents and grandparents are probably looking for. Certainly those are the kind I'm looking for. In my day, we earned those rips and those worn out spots. But now you can buy them as if they're already been worn out. In any case, if you go into the store and you have different types of pants or blue jeans or jeans that you can buy, that would be like alleles. Alleles are different versions, the variety, okay? Different ways of doing it, okay? You could think about it as going to a restaurant and they ask you, so you order a hamburger, and they might ask you in some restaurants, would you like French fries or potato chips? Okay? That is a category, a starchy kind of food category, and your choices are French fries or chips. That's 2 versions of what you can order with that meal, with your burger. Okay? So now you got to go back and remember, when you were in high school probably, what term did we use when both alleles for the same gene, both alleles are the same? Okay, so for this gene, on the paired chromosomes, you'll have the potential for 2 different versions of that gene. Okay, and what is it? What's the term we use when both alleles for that gene are the same or that they're different? And the answer is homozygous and heterozygous. So if the alleles are the same on both chromosomes for that gene, then they're homozygous. But if the alleles are different, then they're heterozygous. Some more basics. The genotype of an organism versus, and you'll see where I'm going with this, phenotype. The genotype of an organism is the genetics of an organism. The genotype of an organism is the genetics of an organism. The phenotype of an organism is the physical expression. And that expression will be influenced by genetics and the environment. So the question comes up, if the genotype is a mixed genotype, it's heterozygous. If, for example, in a flower, there's one allele for white and there's one allele for red, what will the phenotype be? And it depends. Some traits simply show dominant recessive. So if we had a white and red allele for flower color, it could turn out that white is dominant. And so even though the signals are mixed for flower color, the flower still ends up being white. Okay. It's also possible that there's something called incomplete dominance, in which in the white flower color and the red flower color, we get something like a pink phenotype. The phenotype would be mixed. Okay. By the way, before we go on, one of the things that's the real advantage of me recording this and showing you the PowerPoints versus in lecture is that you can go back and play this back and say, wait a minute, I wasn't paying attention. I got to go back and hear again what he said. And so I'm really pleased that this is recorded for you. We don't have a chance for the interaction. I hope all of you feel free to email me and ask me about something. And remember that the day or two just before the test is not a good time, and I may not get back to you in time with a sufficient explanation if you wait until a day or two just before the test. Okay, so I'm delighted that you can play this back, especially on chapters 23 and 24, which are a bit more technical. Okay, now some bigger terms that you may or may not have addressed in high school. The gene pool. Of course, we've had an awful lot of fun thinking about a swimming pool here and getting into the gene pool and getting out. And no, you don't need a towel if you've visited the gene pool, okay? So there's lots of jokes about that. And now I've got those, I hope, out of the way, and let's go forward. So what is the gene pool? It's all of the possibilities for all of the genes in a population. Boy, is this big, and it's defined as well in your textbook. So what it says is, in this population of red fox squirrels in Carlinville, anywhere, somewhere in the red fox squirrels in Carlinville, is there an allele for albino squirrels? Or maybe it turns out that no, not at all. Now, some of you may know that in Olney, Illinois, I believe it's O-L-N-E-Y, we had a student from Olney, Illinois. He was a very, very friendly student, and sadly, he died. And I was very close to him. We played racquetball together, in fact. Well, in Olney, Illinois, there were albino squirrels, and there's just a big population. And so the alleles for albinism are very abundant in the gene pool of these squirrels in Olney, Illinois. But maybe there isn't even a single squirrel that has a single allele for albinism in Carlinville. I've not heard of albino squirrels arising in Carlinville. It must be pretty rare. Imagine, by the way, if you wonder why albinism might be rare, if you have to have both alleles for albinism, and it turns out, let's say for deer, I don't know these numbers, I'm just making it up, but let's say that 1 in every 10,000 deer in Illinois carry one allele for albinism. Well, 1 in 10,000 deer needs to meet the other 1 in 10,000 deer to have babies. And then the way this works is, on odds, only 1 out of every 4 of their babies would be an albino. So that's pretty rare. But what if the allele for albinism was really abundant, as it is in these squirrels in Olney, Illinois? And so I don't know the numbers, but what if it turns out that almost every squirrel in Olney, Illinois has at least 1 allele? Well, if it doesn't have 2, it probably would be dark. But if it got 2, or its babies, or the babies that were produced, then they would be. Okay, so we wonder about the frequency of alleles, the possibility of certain traits, certain phenotypes emerging because alleles for that trait are found in a population. These are the kind of things that we wonder about. Okay? Let's leave it like that. So what is microevolution? Well, microevolution now, in one way to define it, in a genetic way to define it, is this change in the frequency of alleles from one generation to the next of a population. So if an allele is found that leads to a phenotype that helps somehow that organism to survive, then we would expect that in the next generation we'd have more babies carrying those alleles than we did before. Well, sadly, and I haven't heard anything about this because I think we're not there yet, but what if it turns out that some of the variation in how people are responding to the SARS-CoV-2 virus is due to different alleles that we have, perhaps different proteins or carbohydrates on our cells? We might be able to track down in the future an explanation why some people don't seem to get very sick from the virus, but other people do. And it may be due to some of the genetic differences. We think that there are other factors as well that affect this. For example, being older and also having trouble with respiratory problems like, emphysema, goodness gracious, all kinds of other respiratory problems, but caused by things like smoking or living in an environment where every day, just go outside and you're breathing the industrial wastes of maybe an oil refinery or a coal burning power plant. And so those people are already beaten down a bit and are more likely to suffer from the disease. But some variation in our response may be due to different inherited frequencies of alleles or the presence of certain alleles in a population. And so if it turns out that this disease stays around, there may be some people who are more likely to survive and reproduce because they had certain resistance to this disease. So what is microevolution building on? It's building on variety that's generated by both mutations and then new combinations of traits, that's what sexual reproduction, followed then by selection by the environment. So this is what we've talked about many, many times before. Microevolution, by the way, can also be thought of as simply the small and gradual changes from generation to generation that are produced by natural selection. So it need not be a genetic definition. It could just be seen as small evolutionary incremental change. So we expect that if there's variation in the genotype of organisms, it can lead to variations in phenotype. Now, some variations in genotype don't produce changes in phenotype. We said in the simple case of the simple dominance, you could be homozygous or heterozygous, but if you had the dominant allele, you'd see the dominant trait expressed. You'd see the phenotype, because you get the same phenotype if it's a dominant phenotype, simply by being heterozygous or certainly by being homozygous for that particular phenotype. Okay. So, sometimes we see lots of different phenotypes in blood types, blood types and Rh factors. Turns out, so these are very, very simple classic cases of inheritance of alleles that lead to very distinct phenotypes. And now my point is that it's not always that way. There is some phenotypic variation that isn't heritable. Okay? Not all phenotypic variation is heritable. And so you might remember I've made reference before to identical twins. And maybe you know identical twins who are in their 20s or even much older. And especially if they're much older, you'll notice that there are differences between them. That when you look at them, you can see differences. And those differences are not just due to genetics, but they're due to other phenotypic variation that isn't heritable. So here's a real focus on that, variation that isn't heritable. So you're reading about Darwin's finches, and the authors were Peter and Rosemary Grant, who I think are still at Princeton University. And what it said on page 381, when Dr. Grant, one of them, I don't remember who, notes that when they looked at beak variation, they determined that, and then this was in your reading, but I don't know if any of you really thought about it. 74% of the variation in beak depth, they could track down to certain genes, certain alleles. What's the other 26%? Well, that's phenotypic variation that is not directly determined by your genetics. And that's environmental causes. I think that when students start learning about genetics, there's a tendency to think that, well, this is it. Genetics is everything. But genetics is expressed in environments, and the environments influence the genetics, and the environment influences the final phenotype. So, we have other places where we see this, not just the thickness of beaks in finches in the Galapagos. Your own human height. And it would be fun if any of you know who these two people are. It's a team that does magic, and they're in Vegas, and they actually have a good TV show now where they bring on magicians to try to fool them. And maybe you know this is Penn and Teller. Penn Jillette and Teller, and I don't know Teller's first name. In any case, clearly we have a tall person and a shorter person. Is that just because of genetics? Well, if you looked, and I could use a picture of my family here, if you looked at a picture of my daughter and my son, you'd see that my daughter and my son are both taller than my wife and I. How's that possible? Well, some is genetics, but there are other explanations. My genetics probably indicates that I should be over 6 feet tall. My dad was 6'3". But about middle school for me, so when I was about 13 or 14 years old, my thyroid gland apparently stopped working, and that greatly affected my growth, my height. And so I am currently my height of when I was in 8th grade. And when I was in 8th grade, I was one of the taller kids. But then, like watching the corn crops grow in the summer, I stayed the same height, and everybody got taller around me. That was my high school years. In any case, for me, my genetics probably indicates a much taller height, but the environment and my physiology affected my expression of my genetics for height. And so I wound up being shorter. For me, that conveys advantages because I'm more comfortable on airplanes and I don't need a big car to be comfortable. I can get by with a Toyota Corolla and be quite comfortable with a little cheaper car. In any case, your height and expression of both your genetics, your health, and the environment. Here's a figure 23.5 in your textbook, in which what they've seen is this particular kind of, and I think it's a butterfly, has a caterpillar here that looks dramatically different. It looks, I think these are oak pollen sacs, or flowers, oak flowers, I think. And so when the caterpillar's on a tree like this with these oak flowers, that's what the caterpillar's body looks like. But when it's on a tree where it doesn't have those oak flowers, it has a different phenotype. And it depends on what they eat. Isn't that something? So what they eat causes then an altogether different phenotype. The genetics, we think, are the same. Oak flower eaters and oak leaf eaters. So figure 23.5 in your book. More examples of phenotypes that are not direct results of genotypes. Okay? So backing off, reminding you, just doing a big picture here. We said blood types are a good example of genotypes dictating phenotypes. What we're looking at right now are genotypes that don't lead necessarily to particular phenotypes. So, in sex determination, males in sexually reproducing organisms, and here we're talking about that 5% or so that are vertebrates, there is genetic sex determination, just like what you're used to. So XX for female humans, XY for male humans. But it turns out within vertebrates, there are some vertebrates in which sex is not genetically determined. Instead, sex is determined by the temperature at which the little embryos are incubated before they hatch out. And so warmer temperatures and cooler temperatures lead to different sex ratios in turtles and crocodilians. And at the meetings that I go to, there are people, including lots of biologists at the University of Illinois, that really are trying to narrow this down. So when you think about the incubation temperature of turtles and crocodilians, think about the fact that it's not just one temperature, right? These things are laid in the ground or amongst vegetation, so gathered rotting vegetation. And the temperature is going to go up and down depending on the day and time or the night time. Plus, there's going to be variation from temperature. So right now, it's spring. And we've had a day where I think it's pretty much close to 80. It's already been last week. And yet, it's also been freezing at night. And so I think 2 days ago, we had temperature that went to freezing. We had to cover some plants. So there's weather variation in temperature, and there's fluctuation just from day to night. And so people are exploring this and finding out, well, so what temperature is it that actually determines whether it's male or female? And is it an average temperature, or is it a high or a low temperature that the embryos experience? And it's not clear. They're working this out. And so, in number 3, we see something that's a combination of both genetics and temperature, and it's influenced. And so this is, by the way, how many things work in biology. So you got your genetic sex determination, you got your temperature sex determination. Oh, good, we figured it out. Well, it turns out sometimes it's not just one or the other. And that's where we spend most of our time in biology. We can quickly get past the things that are simple, and then we spend all our time trying to figure out the things that just don't easily fall into one category or the other. Now, another aspect of traits has to do with, and maybe you're aware of the terminology or not, but there are terms that we use in reference to numbers that are proper and improper. So, is it proper to say that there are less people in my class than last year? Or is it proper to say there are fewer people in my class than last year? Fewer has to do with distinct groups. Okay, so I don't have 0.287 of a human being. I either have a whole human being or not in my classes. And so we use the word fewer. But if I asked about weight changes, so since you've been staying at home, are you heavier or lighter than before? Okay, that's a matter of, do you weigh less? So fewer people weigh more, maybe because you're all staying at home. So we have these different kinds of traits, things that are sort of black and white combinations and things that are matters of degree. Qualitative characters are like people. They're either there or they're not. They're very distinct. Okay, so I either have a person or I don't. Okay. In biology, there are some intermediates between male and female. But generally speaking, you can think of these as male or female. It could be pregnancy. So the females are either pregnant or they're not, but they aren't partially pregnant. Okay? Blood types fit into this category. You're either A, B, or O. Sorry, that's your choices. Quantitative characters are like the weight that I was talking about. So people may weigh less or they may weigh more. So I said in my setup here was when you're talking about people, it's fewer or more people. But when you're talking about weight, for example, it's less or more. These are Ers. I'm trying to help you out here. This is the way I explained it. I don't think this is the way it's in the book. So I think of qualitative characteristics as Ors, quantitative as Ers. Ers are matters of degree. Ers are matters of degree. You're faster, you're slower, you're taller, you're shorter, you're darker, or you're lighter. Okay. And you, of course, can go back. That's the beauty. So I'm not so worried about whether I'm going a little too fast. You can just go back. How about the explanations provided by Mendel and Darwin? Were they looking, was one of them looking more at qualitative and quantitative, or the other? Well, what did Mendel do? Mendel looked at pea traits. Peas were either wrinkly or smooth, flower colors white or pink. These were qualitative. These were discrete. These were traits that fell into distinct categories. Traits that fell into distinct categories. Okay? And you're going to learn a lot more, you probably did in high school, about Mendel studying these traits, mostly in garden peas, but other plants. Darwin, see in his own descriptions, and you've already read then, Origin of Species, or parts of Origin of Species, Darwin was much more concerned with quantitative traits, like rabbits that are faster or darker. So Mendel studied Ors, Darwin studied Ers. Remember that Mendel did his work at the time of Darwin's life, and that Mendel knew about Darwin's work. We don't know if Darwin knew about Mendel's work. Mendel's work was not widely publicized. And so in the early 1900s, again, Dobzhansky, Ernst Mayr, who gave us the biological species concept, Dr. Ernst Mayr at Harvard. For many years, when I was teaching this class at Blackburn, Dr. Mayr was alive. I got to be at a meeting with him, got to sit right near him. It was really quite a highlight. In any case, in the early 1900s, so about 100 years ago, when we rediscovered Mendel's work, remember Mendel studied ores. A pea seed was either wrinkly or smooth. And Darwin studied urs, bunnies that were better camouflaged or faster. And so there was a potential conflict. What was this? It seemed like the genetic explanations were categories, and yet Darwin was seeing matters of degree. Well, that was thought to be a conflict earlier on, as we were working through our basic understandings of how genetics works. Now we know it isn't a conflict. We know that genetics and evolution act on both types of traits, qualitative and quantitative. All right. What are polymorphisms? Well, just do your word roots. Poly means it's a parakeet, right? No. Poly means many. Morph, shapes, so many shapes. Polymorphisms, variations of a trait. So blood groups are good examples of polymorphisms. We have these different kinds of phenotypes based on variations in genotype. So the single trait, our blood type, varies. It's a good polymorphism. So what we can do in population genetics is we can look for variation in alleles. We can look to see where there are differences. Okay? Remember that differences are what's going to matter for evolution. So, take a look at this definition. I believe it's also in your chapter. Geneticists can come in to a population and they can look at a particular trait, and they might find that for all of the organisms that live in that population, they all have the same allele. And that's said to be fixed. It means then that for that particular gene, there is no variation. Everybody in the population has got the exact same allele. Go to the restaurant, you're always getting French fries with that. There is no chance to have rice or to have mashed potatoes or chips or whatever else it is they might serve for you. Okay? So for a particular trait, it could be fixed. On the other hand, there could be variation. And so geneticists are interested in, on average, how much variation is there where there's options. So a gene is found at a particular point along the length of a DNA molecule, along the length of a chromosome. Okay, so a chromosome is a DNA molecule with the packing materials, with proteins. So a chromosome, it's got a single DNA molecule in it, and somewhere along the length of that DNA molecule is a gene, and that's the locus of that particular gene. Now think, if you want another analogy, and I always like my analogies, they help me, I hope they've been helping you. Think about a train with 100 cars, okay? And it may be that the 30th car in the train is a tanker, and it's carrying ethanol, maybe to mix with gasoline. So the loci, the location of that particular tanker, is the 30th car in the train. Likewise, there's a locus for a gene along the length of a DNA molecule in a chromosome. Okay? So, when we have two of those chromosomes, which is 2N, right, diploid, then we have two chances for this gene. And that gene can vary between the two chromosomes and be the same. That's homozygous or heterozygous. Okay? So the variability might exist where we have that gene. And I already did the restaurant menu. Okay? So this is pretty much what I've just been saying. I was just ahead of myself on the slide here. So if there's no variation, the gene is fixed. And just like I said, you're always getting fries with that. Boy, I've been doing this a long time. So what we can do is we can actually look at the individual nucleotides along the length of a DNA molecule. We can look at those nucleotides along the length of a DNA molecule and see if there's variation. So now when we're looking for variation, we might find variation within a population and between, just as it says. Between populations, this is geographic variation, because these populations live in different places. Between populations, if we find differences, then movement of individuals between populations allows these two populations to start sharing some of the variation. So if there's more variation in one population, and individuals from that population go over and have babies with individuals in the other population, then that population will get some of the variation from this population. So there's variation within a population that matters, and it also matters that there's variation between, and that variation has a chance to spread. This, by the way, the spreading of this variation between populations is a real important principle in conservation biology. It has to do with some major changes that we're trying to make to our national parks, our state parks, and all sorts of areas that have been protected. Because if we create these parks and they're isolated from each other, then there is not a chance for movement between the populations, right? If they're like islands, so if Yellowstone National Park has wolves, but those wolves never meet up with other wolves from farther north in Canada, then there isn't the sharing of the genetics. So what we're talking about here is not simply about evolutionary biology, but practical applications to conservation biology. Okay. Well, here's a figure in your book that shows you how much populations can actually vary. Here, this is, of course, an invasive species, pretty much true about all sorts of these European mice that were introduced to this island. And in the different ends of the islands, oh my gosh, not just allele variation, but even the shape of the chromosomes. I'll let you look at that. You can also look at it in your book in a little bit more detail. Interestingly, and this is the part that's a big surprise, is that, and I'm seeing that this is in the 9th edition. I'm not sure that this is in your current textbook. They may have eliminated the figure. But in any case, back in the 9th edition, when this was in the textbook, it was really fascinating to see that not only was there allele differences, but even the shapes of the chromosomes. And yet, here's what they found, that it didn't seem to affect gene expression. I can tell you that sometimes it does. And that could be medically significant in a human. All right. So we're talking about differences between populations. And what we're seeing is that geographically, that different populations of the same species may actually have different frequencies of alleles because they're experiencing slight variations in the environment. Okay? And so along a continuation, so it might be going up a mountain from lower altitude to higher altitude, that those populations living at higher altitudes actually do face cooler, right, cooler temperatures. They're also going to face less oxygen. And so these series of populations that are sort of lined up over a subtly changing environmental variable like oxygen or temperature, those variations, that continuous variation, forms what's called a cline. And so here's a flowering plant that's going up a mountain slope. And so I want to make sure you can see what's going on here. My understanding is that this would be Pacific Ocean over here on this side on the far left. And then you can see this is this line and these dots. The dots are the study sites. And the line shows you the altitudinal change. And so here is altitudinal change until you get really, really high up. And that's going to be wetter on this side of the mountain, by the way. And then it's going to get much drier. Okay. I'll just tell you that I actually drove and got to be in this area. So about over here is Las Vegas. And you can fly into Las Vegas, rent a car, as we did. Then we drove to Southern California. And then we came back, actually way over here. Well, and I think we may have stayed in Mather. In any case, then we made our way up to Yosemite and Sequoia National Park and crossed over the mountains and then came back to Las Vegas. Death Valley is also down here at that same kind of level. Well, with this variation in altitude and variation in moisture, what you can see is this one species of plant has all of this variation. And this, I think, is the height, 75 centimeters height. And here you can see gradually, and that's what makes a cline, gradually we see shifts in a phenotype from one location to the next as we move all the way across here. And now to this dramatic change, drop in altitude, drop in moisture, and now we've got very, very small plants here. So this is a cline. This is a gradual variation of a trait over a continuous area where we have different changes in the climate as well as altitude. Here's another kind of variation in an allele. So there's a specific allele. Remember, it's a version of a gene. And the gene that codes for this enzyme functions better, they say, at colder temperatures. And so in this particular fish, here it's living in Maine that's very cold. And so I think this is the frequency. No, that's latitude. Sorry, that's latitude down here. Here's the frequency up here. So in Maine, at this much higher latitude, okay, which means it's farther north, it's very cold at farther north. And here, the allele that codes for this enzyme, oh my gosh, everybody's got it. And now as you move all the way down to make your way to Georgia, the allele becomes less frequent. And so we're down here with the allele frequency for being less than half. So that's what 0.4 means. 0.5 would mean half. And so all the way down here you almost never see even the allele down at these warmer temperatures. So this is an allele that's helpful for living in a colder environment. And so in populations of this species, it's very, very high. And then as this species extended its range all the way down here, or maybe it started here and went the other way, here that particular allele is not so useful at these locations that are warmer, and therefore that allele is less abundant in that population. So these are differences between populations of a same species living in different places. And that's a cline, a cline. So, genetic variation. We've seen genetic variation within a population, like blood types, and between populations, both the flowers on the mountain up above and also that allele for those fish. So the next question is, how do these variations arise? There you go. That's what we've been talking about before. That variation, genetic variation, arises simply because of mutation. Now you get something new, or simply recombinations, sexual reproduction, like this. So this is like dealing cards we talked about before. So playing poker or something, you're getting different cards. Here, it would be like a new type of card. I don't know, an ace of squares. Okay, so we got diamonds and we have hearts, so maybe we should have an ace of squares. Well, that would be a mutation. That would be a new type of card. But if we don't make new types of cards, we could just shuffle the cards, and that's what we do when we're playing card games, and that's what sexual reproduction does. Okay. So let's talk about mutations. What are they actually? Well, it's this change in the nucleotide sequence. That's what it is. Okay. So in DNA, we have nucleotides arranged very precisely, and that's what the information is that's going to help determine proteins. And when we have mutations, they could cause new genes and new alleles to come about. We're going to see, by the way, that some mutations actually don't cause any change at all. That's kind of neat. It's a kind of a silent mutation. It's important for you to know that mutations that arise have to be in the cells that produce gametes. And so you might remember that all of the cells in your body belong to one of two categories. They are either somatic cells, which are the cells that are not making eggs and sperm, or sperm. And so it's the cells that are in your arms and your legs and in your head and in your chest. Those cells that make muscle and nerves and skin, eyes, for goodness sakes, all the special cells in our eyes, those are all somatic cells. Mutations need to arise in germ cells. Those are the ones making eggs and sperm. And so mutations there will be passed along to the next offspring. If you, and most of us do, go out and expose your arms to a lot of sunlight, that sunlight will lead to mutations. And those mutations will cause changes in our skin. They may cause freckling, or they may cause skin cancer. And so we have to be very careful about the exposure of our skin to sunlight because it could lead to cancer. Those are mutations on your skin. But those mutations are not going to be passed along to your next generation because those skin cells are already separated off from the cells that are making eggs and sperm. All right, so that's the somatic cells and germ cells. And sunburns that we talked about. And we said no, those will not be passed along. Now, point mutations are very, very specific. It's causing a change in just one base. It's not a radical change. It's just one, well, we sometimes say letters, but it's a nucleotide. It's a letter that represents a nucleotide. So G, C, A, and T, those are the nucleotides. And we use those letters to represent those nucleotides. A point mutation is then a change in just one of those letters, one of those bases. All right, so what is going to happen when we change a base? Some mutations, as I said already, actually don't have much consequence, and we'll see how that could be in just a few minutes. Some might actually produce a redundancy, some mutations. So there's 64 different codons. Maybe you've learned that before, and they code for just 20 amino acids. We talked about those 20 amino acids when we talked about this in zoology. Okay? Here is a fine example of the codons that are coding then for proteins. And we can see, and there's a way to do this. So the first letter, they set these up differently sometimes, but in this one, here are the bases, UCAG, down on the side, and then likewise across the top. And so what is of the triplet of the codon, what's the first letter? So let's say it's going to be C. What's the second letter? And let's say it's going to be C. And so this square represents codons that begin with C, and the second letter is C, so right here. And so what is it that's being coded for by C, C, and then something for the third base? The answer is it's the amino acid proline. So here, imagine a point mutation that causes C to switch to A. Would there be a different amino acid coded? And the answer is no. Now, these are, by the way, of this codon, these are RNA now. This is not DNA because you've got uracil there. So here we're taking a look, making sure you understand that. Here, if there was a switch from C to A or C to G or any switch between any of these, it has no consequence. It still leads to the production or the matching or the information signaling, really, the amino acid proline. Can you find a case where the third code would have an impact? So look around at the chart and see if you can find a place where the third code would make a difference. And yeah, there's several. So right here, if it's CA, well, if it's CAC or if it's CAU, you get histidine. But if there's a mutation that leads to either C or U becoming A or G, now you get glutamine. Oh. And over here, it's UU and then UC gives you phenylalanine. But if those are mutated to A or G, you get leucine. And of course, some of these mutations, like here for tyrosine, could be mutated and changed to a stop codon. And oh my goodness, that would make a big difference. So the effect of point mutations, as it says, can vary. It can result in something that's very harmful. It can result in nothing. And it can actually increase, potentially, the fit between an organism and the environment. And those are the kind of mutations, turns out that they're very rare. Mutations can be helpful, and they give us something new, and it may convey an advantage that allows an organism to be more likely to survive and to reproduce. So here's figure 1726, showing you what goes along with sickle cell anemia. A single point mutation leads to the disease. It's just amazing. And yet, as we talked about before, it actually conveys an advantage. So here we can see what is more common, the healthier condition without sickle. Here we can see the change. The change leads then to these differences in the messenger RNA. And that code is for glutamine, and I think this is valine down here. And so that's a single amino acid shift in this protein right here that leads to sickle cell anemia. Sometimes there are mutations that do this. They either delete or screw up or even rearrange the loci of different kinds of genes, the location of those genes on a chromosome. Sometimes even large segments of a chromosome are duplicated. And these kinds of changes, very disruptive like that, are usually harmful. But interestingly, there are some duplications that convey advantages. By the way, this is also an explanation for why genomes can be so large. Duplicated genes, so you used to have one and now there's a second copy of that gene on the chromosome. That duplicated copy can take on new functions if there are additional mutations. And that could be really, really important. Okay? So duplications of a gene, if it happens, and this, by the way, is not rare, we know that there's been lots of cases of this. A second copy, when you have this duplication, can undergo mutations that would change the original function, but it might allow a new or related function to arise. Now, this idea of copying something and then playing around with the copy, but not hurting the original, is something that, well, we do this all the time. If you've ever had a chance to hold in your hand a copy of the U.S. Constitution, you could look through it, you could mark it up, you could highlight it. Heck, you could have even had a test over the U.S. Constitution, and you might have even been allowed to bring in a copy of it. Okay? So you might mark it up. But you aren't going to bring in the original Constitution. Okay? Well, there's some people who are guarding the Constitution who would probably stop you, and none of us would want you to have an original copy. Okay? We're going to keep that in the very safe places. The National Archive, which is located in Washington, D.C., in very, very special place. But we have copies of the original U.S. Constitution, and that we can mark up and edit. You might do the same thing when you revise a paper that you've been writing. I hope that you have a paper and that you've had like a copy that you had last night. This was the best draft that you had last night. And now when you wake up in the morning, you should go through and edit it. It is something that I do all the time. But when I edit it, as soon as I open up the latest draft, I save it under a new name so that I'm editing, but I'm not changing the draft that I had last night. I've saved the draft from last night, and now my edit is going to be of a second copy. Okay? That's the beauty of this gene duplication. And I'm going on and on about this because this is huge. And this is something that we've discovered only in the last few decades because we've really gotten in and learned much more about the genomes of organisms and crucial changes like this that are important to evolution. All right, how about mutation rates? Just how often do we see changes? Interestingly, they're relatively low in animals and plants. Here, a mutation in about every 100,000 genes per generation. Interestingly, they're even lower in prokaryotes and viruses. But because these organisms, and a virus isn't really an organism, so we have to figure out whatever we're going to call a virus, because it's not really a life form, because it can't reproduce without another host organism like us that has regular mechanisms for copying DNA. So viruses cannot replicate their genetics on their own. They have to have a host organism. But in any case, the replication of viruses and of bacteria is so much shorter lifespans that they go through so many more replications, thousands, perhaps millions compared to a human lifespan. So even though the mutation rate is less, they do accumulate because they replicate often. So I was talking to you already about the SARS-CoV-2 virus that we're seeing, and they're seeing in these viruses in New York City, and I guess they have mutations that identify them most closely as coming from Europe, so we can even trace that viral ancestry like that. All right, so another aspect of this, sexual reproduction. We said that's like shuffling cards, right? And here's a real important statement, really important statement that's in the middle. Sexual reproduction is more important than mutation in producing the kind of variability that makes adaptations possible. Okay. So, if you're going to ask the question, what is the variety that's going to allow us or allow goodness, a frog population that's facing a fungal infection. What kind of variety is going to allow these organisms, us, frogs, to survive some kind of threat? The answer is combinations of existing traits, not mutations that convey some big new difference. It's mutations that occurred long ago that provide variability now, existing variability, that simply is reshuffled. That's what this is saying. That what is it that's most important in making immediate adaptation possible? It's reshuffling of existing traits. Okay? So that's really important, and you have this on a video here so you can look at it again. All right. Mutations, by the way, just to go on and explain that. So I'm going to go back the slide just to explain this. Mutations, if they arise, so right now in humanity, if all of us are, and we aren't, that's good, but if we were threatened by some kind of viral infection that was really devastating, and that a new mutation, if it arose, let's say, in a population of people that live in Haiti, okay, the country of Haiti, if there's a new mutation that conveys resistance to some new global viral threat, well, there will be some lucky people in Haiti. But the rest of us won't gain from that mutation. That's why mutations do not, the arising, the origin of new mutations is not the main way that organisms are going to survive from one generation to the next and have much impact, because the mutation isn't widespread in the population. Now that's my bottom line. Mutations, when they arise, are very limited to a few individuals, or maybe one individual. And it's going to take many generations for that particular allele, that new kind of allele that arose through a mutation, it'll take many generations for it to spread throughout the range of that species. For us, it's all over the world. So, a few lucky people in Haiti that have mutations that convey resistance is very fortunate for them, but it doesn't help all the rest of us, because we aren't going to get that allele. Our populations aren't going to see that allele for probably many, many generations in the future. All right, now back to sexual reproduction and how it generates diversity. And I'm going to go a little quicker here because you can go through the book and learn all about these, and because we already talked about them in zoology. So independent assortment, you got to remember what independent assortment was. And there's your hint, showing you a pair of shoes. Now that's not the pair of shoes I showed you in zoology. Independent assortment is the question of, well, I have to back up. When an individual, a human, let's say, is going to make eggs or sperm, you are not going to give in the egg or sperm all of your genetic material. Instead, you're going to give one member of each pair of chromosomes. So one chromosome from each chromosome pair will be passed along in an egg or sperm. Independent assortment is asking the question, which member of each pair is going to be passed along? And the answer is, it's just random. It is truly a random assortment of which, so here I used right shoe and left shoe, which shoe, which member of each pair is going to be passed along in an egg or a sperm. So you can go through this again. I see this is from 10E, but the same figure is in 11E. It may not be that exact figure number, but it'll be really close. And this is showing you, just to remind you, chromosomes that have already replicated their DNA. So this is one chromosome that's replicated its DNA, but those two DNA molecules are still joined together. There's often confusion in this for students. So here's one chromosome, there's another chromosome. Each of these have replicated their DNA, okay? And these are sister chromatids that are still joined together. when these pairs of chromosomes line up like this in meiosis, when these pairs of chromosomes line up, okay, they could line up this way with all the reds on one side and the blue on the other, or they could line up this way. And so in the future, this side would sort over here, this side sorts over here, and here you have a red and blue, or a blue and red combination. And over here you have just blue and just red. So they're showing you with two pair of chromosomes how we see independent assortment causing variety. And so we went through this in zoology. How many different combinations of chromosomes can exist in a single egg or in a single sperm? How many different possible combinations are there just due to independent assortment? And the answer is this 8,338,608 different combinations in eggs or in sperm. And now when you combine this together, if you can have 8,338,608 different eggs or that many different sperm, then what are the different combinations of eggs and sperm that you could get from a man or a woman? Just independent assortment. How many different kinds of zygotes can you produce? And the answer is, if you square this number, it's about 70 trillion. And I made a fuss before in zoology, you might remember, that a lot of textbooks simply say that there's about 8 million different eggs or sperm, not 8,338,000. And so they square 8 million and they write that there's about 64 trillion different kinds of zygotes that could be produced. Well, the real answer is 70 trillion, not 64 trillion. And you might say, oh, come on, Zelisko, why are you fussing? The difference is 6 trillion. Okay, so I kind of think we should reuse this number. And so the answer is closer to 70 trillion. So, just by independent assortment, if your biological parents wanted to produce, just by independent assortment, and give the same chromosomes to another sibling of yours, to make another brother or sister, they'd have to have 70 trillion babies. And that's just by independent assortment. Reminding yourself that we have about 330 million people in the United States. There are about 7 billion people on Earth. Okay, 7 billion times 10,000 is 70 trillion. So 70 trillion is 10,000 planet Earths today for the population of people. So that's a lot. Well, we also went over the fact that genetic variety, sexual reproducing forms, is not just independent assortment. So 70 trillion is just not enough. Because we said before independent assortment occurs, there is some exchange of genetic material. So before we had chromosomes up here that were red and blue. Now they're red and yellow. Here's all the red one that it's replicated, so it has a sister chromatid red. Yellow's done the same thing. And instead of just pulling apart the yellow from the red members of this pair, they exchange. They swap pieces. That's crossing over. Okay? They swap pieces right here before they sort out these chromosomes. And that means these are the chromosomes you inherited, the yellow or the red one. And what you pass along are not the chromosomes you inherited, but new combinations. For each pair of chromosomes, you pass along a chromosome, a single chromosome, that actually is a combination of mom and dad. A combination of what you got. So you don't pass along just what you got. You pass along a combination of what you got from mom and dad. So what we did, just by comparison, I think it was Harry Potter, if I remember right, in zoology. But now, imagine that you're watching Bambi and Shrek. Okay. I'll go back. And you're watching Bambi and Shrek, and you've watched 4 minutes of Bambi, and now suddenly you're going to watch Shrek for 1 minute. And then after that 1 minute, you go back to watching Bambi. Okay. Likewise, you're over watching Shrek for 4 minutes of the movie. You're just getting settled in your seat, probably throwing that popcorn in your face. And all of a sudden, you get Bambi. Bambi for a whole minute. And then after that minute, you go back to Shrek. Goodness, somebody could have gotten up to go to the bathroom, or somebody could not even be there, and you're trying to explain this to them, and they're going, "Oh no, it's Shrek. See, it's Shrek. What are you talking about, Bambi?" Or the Bambi people go, "What are you talking about? It's a minute of Shrek." That's the swapping of the 5th minute of the film here. This is an analogy to what's going on over here. Okay? It's a segment of these chromosomes that gets swapped out. All right, so 70 trillion wasn't enough for us. Crossing over is now an exchange of pieces of the chromosomes. Crossing over is nearly random. It's on average 2 places for crossing over. So, oh my gosh, this might be 70 trillion squared. I mean, the number is so big that it doesn't really matter. It's beyond anything we can imagine. And then finally, which sperm and which egg are actually going to come together. So there are some parts of fertilization that are truly random, and then there are some parts that aren't. And the fitness of the very individual sperm might have to do with its ability to make its way up a female's reproductive tract and be able to fertilize an egg. Some parts are random, and so if we take a look at, in many cases of vertebrates, where the males in the breeding season are simply out always looking for a female, and any female at any time that's somewhat receptive, it's a chance for a mating opportunity. In humans, there are times when women are more fertile than others. There are times when they are more likely to get pregnant. That's what I mean. And so it's possible that copulation occurs, but that particular sperm are going to make it up to the right part of the oviduct, but lo and behold, there's no egg there. So we talk about this in development. I talk about it in my human reproductive biology class. But it's not the fastest sperm that wins. It's shown that way regularly in TVs and cartoons and other movies. You can be really, really fast, but if there's no egg there when you arrive, well, then you don't lead to fertilization. So you can go out right now today and run a marathon, but you won't win the race. You're going to win the race only when they're running the race. So wherever the Boston Marathons run and wherever the finish line is, on any given day of the year, you could go run that route. And if you're the first one to cross the place where there would be a finish line, you can be really happy, but you didn't win the marathon because they weren't running a marathon, okay? So if there's not an egg there, you can be as fast as you want to be, but it's not going to make a difference. So some parts are random. Was mating correlated with ovulation? And other parts have to do with the fitness of the gametes, the fitness of the eggs and the sperm. So, holy cow. Just because of independent assortment and random fertilization, I mentioned this already, there could be independent assortment, 70 trillion times 70 trillion, different offspring from a single pair of parents. So that should be 70 trillion squared, I think, from different offspring. And that's just a guess. But it's an astronomical number, so much greater than 70 trillion. Add crossing, oh, sorry, that last one was for without crossing over. And now that's my point. I've squared 70 trillion, and I don't even know what this number is. So millions, billions, trillions, quadrillions, I don't know, pentillions, sextillions, septillions, octillions, who knows if that's even how they number. 4 octillion, I don't know. This is where in chemistry scientific notation is very, very handy. All right. Now starting something else altogether, and that's Hardy-Weinberg. And I think what I'm going to do is stop here, and then we'll go through the Hardy-Weinberg and the rest of chapter 23 with the next lecture. I'm also going to get myself another cup of coffee.